This video shows the R1 representation of Connectron loop structure. This is a rather complex graphic and but very powerful. As I rotate the image around, you can see that this is basically a huge collection of spirals. This is the King software, and so there are a number of different switches that I can turn on and off. So this spiral in the center, which is called the whole chromosome, well, this is mouse chromosome 1. If I put on, for example, the gene coding switch, then these are all of the connectron loops for the various types of connectrons in chromosome 1 that are caused by gene coding transcription. The switch, the set of switches here determine which section of the structure you're looking at. So you can look at just a very small portion of the structure and see just a very small collection, portion of the total chromosome. So this is very powerful. And then, of course, if I turn off the gene coding and turn on the non-coding, then you see that these are the connectrons that are produced by the non-coding events in the mouse chromosome as a function of the connectron determination program. So let me simplify the presentation by just showing you a set of just a few of the connectrons. You can play with this yourself when you have King and the different graphical data sets. So here we have the complete chromosome, and we have the so-called one-shot connectrons that are for the gene coding and the non-coding. So this is the gene coding, and these are the non-coding. Okay. Now, a one-shot connectron is one where when a transcription occurs, the effect of that transcription is to turn off the transcribing element itself, whether it be a gene coding or a non-coding element, and to turn it off the transcription for the lifetime of the connectron, which is determined by the length of the triple-strand helices that are formed as the RNA from the transcription event wraps itself around in two places, the major groove of the DNA of the target. So what we see here are two different sets of connectrons, but of the one-shot variety. And so then there are others. There are a whole bunch of other different types of connectrons that I'll talk about in a few moments. Well, let me turn off the non-coding elements for the moment and focus in on what perhaps is the most interesting aspect of the connectron structure of the mouse. So let me pick a particular place. Let me choose this thing right here. And now we'll zoom up, and so you see that here's a loop that starts at position 101. There's 101 million bases, 0.1, and no, it starts at 99.5 and goes to 101. So it had just the opposite. But you see, in this particular place, there's only one rule, number 3782, that it describes the transcription event of a gene coding sequence, and that then produces this loop that is well, 99 and 101 is about 2 megabases long. This is characteristic of many places, and so we can pick a place over here, for example. I guess I have to go there, and here you see to other ones that are where there's just one. Now, if we pick this place here, you see that, well, there are actually two there, but this one is only a very short loop that's formed, whereas here this is a much larger loop. Now, there are other places where, for example, here, where as you zoom up, you can see that there are quite a number of different loop structures formed by transcriptions from a number of different chromosomes, possibly in the mouse genome. So again, there are some that are relatively short, and some that are all relatively long, but with differing endpoints. So each of these connectrons, each of the loops formed by some transcription event of a gene coding nature, try to turn off the transcription of whatever genes or non-coding events are in this region here. So we'll back off again and center like this. And so now you see that we'll turn on the gene coding events and turn off the non-coding. And once again, you see that there are a significant number of connectrons formed by the non-coding elements in the mouse genome. Once again, I'll pick a particular dense region, let's say here, and zoom up on this. Now this is just the non-coding connectron loops that are formed in this particular region, and then here are the gene coding loops that are formed in this region. So what you see is that the connectron structure of a genome is actually quite complex. So it means that there are, well, perhaps this number is 50 different blue lines here. There are 50 different transcription events throughout the genome that attempt to turn off the transcription in roughly this area here. Another one tries to turn off a much larger area, or will turn off a much larger area. But you see that there are a number of different sequences here. Some have common boundaries and others have other common boundaries that attempt to or will turn off the transcription by forming a loop in this region. But then in addition to the one-shots, then there are connectrons that go, they're on the same chromosome, and they're the primary source of connectron behavior. So I'll turn off the one-shots for the moment and turn on the intra primary. So here you see again a different set of connectrons. Again, you see in some cases they're just one or two. Here there's a whole bunch of things, and you can inquire as to the position and the rule that formed each of these things. But the point is that the spiral gives you essentially, in 3 dimensions, essentially an infinite graphical representation space, and that all sorts of structure can be put into this. Again, this is the intrachromosomal primary connectrons caused by the gene coding and the non-coding. And then there are intra-secondary. We can turn off the others here, and we go on and on like this. So this is a very powerful tool for representing the structure of the connectron structure of a chromosome, in this case of mouse. Thank you.